Hello, welcome to study session, and today we are going to be learning about cells. What is a cell? A cell is the smallest unit of life. It is the basic structural, functional, and biological unit of all known organisms. Now think of this as a house. If the house is an organism, then the bricks that make up the house would be the cells. So cells are basically the building blocks of a living organism. Several cells together form tissues, several tissues together form organs, and then organs form organ system, which finally forms an organism. Now let's discuss about the types of cell. So there is essentially two main types of cell based on their structure and functionality. The first one is prokaryotic cell and the second one is eukaryotic cell. Now prokaryotic cell is essentially bacterial cells, whereas eukaryotic cells can be fungi, plants, animals, etc. Now the main differences between prokaryotic and eukaryotic cells are that prokaryotic cells have no membrane around their nucleus, whereas eukaryotic cells, the nucleus is membrane bound. In prokaryotic cells, the cell organelles do not have any membrane around them, whereas in eukaryotic cells, they are membrane bound organelles. Prokaryotic cells have a single circular chromosome as their genetic material, whereas the eukaryotic cells have multiple linear chromosomes. Cell wall in prokaryotic cells is made up of peptidoglycan, whereas in eukaryotic cells, if cell wall is present, for example in plants, they are made up of cellulose, whereas in fungi, they are made up of chitin. Now cell organelles. This is typically, the model, the one that we are looking at is a model of an eukaryotic cell, and this one is an animal cell. A plant cell is again a bit different from an animal cell in certain perspectives because a plant cell has a few extra organelles in them. For example, they have got chloroplast, which we are going to be discussing in here. They have also got a large vacuole filling up the central space, and they have also got an extra cell wall present to them. Now, cell organelles are like the smallest functional units of a cell, and all of them, they have different functions which together form the cell. And as you can see in here, the different cell organelles that have been labeled. So there's mitochondria, there's nucleus, ribosomes, centrioles, peroxisome, plasma membrane, smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi apparatus, and cytoskeleton. Now we are going to be studying about some of them in detail soon, but I would like to discuss that the plasma membrane, it is the membrane of the cell. It is the material in which actually the cell is encapsulated, and the plasma membrane has a bilipid layer. Now in plants, they have a cell membrane or a plasma membrane, but in addition to that, they also have a cell wall, which is made up of cellulose. And this cell wall makes plant cells a lot more rigid than animal cells. And also animal cells have, as I have said, chloroplast in them, which also we are going to be reading in detail later. And the cell, it has the the substance that fills up the cell, the fluid, is actually known as the cytoplasm. Moving on to this simplified model of a nucleus in a cell. So basically the nucleus actually forms like 10% of the cell. And if we look in detail, the nucleus actually has two membranes around it. So it is encapsulated by two membranes. These are known as the nuclear envelope, and it is double layered. As you can see, the two orange layers are the two different layers of the membrane, and the black space in between is actually the perinuclear space. Now this perinuclear space actually separates the two membrane layers from each other. Now inside a nucleus we have got a nucleoid, which you can see here is the green circle, and then there's the chromatin. Now chromatin, it is formed of histone proteins and DNA strand. So double-stranded DNA, it actually coils around the histone protein and forms strands of chromatin. And this chromatin is linear, but then when DNA replication happens, which can happen during meiosis or mitosis, the chromatin actually coils and it thickens and condenses to form chromosomes. Now this is the part where the genetic material, the genetic information is stored safely in the nucleus. So here all of it is labeled. To go through it again, there's the chromatin, which is in blue, the nucleoid, which is the green circle, the perinuclear space. The nucleoplasm is actually again the fluid that fills up the nucleus, and there's the double-layered nuclear envelope. Moving on, we have got mitochondria. Now mitochondria is a very important cell organelle, and as you can see, the structure, it has got two membranes, and the outer membrane, as you can see, is smooth, but the inner membrane, it is folded inwards and forms this kind of structure which is known as a cristae. And that is inside the cristae we have got the matrix where all the functions of the mitochondria takes place. So a mitochondria, structurally it is rod shaped, it is filamentous, and it is 0.5 to 1 micrometers in diameter. And the mitochondria is actually the outer membrane of the mitochondria is normally smooth, but the inner one is extensively folded inwards to form cristae. These two membranes have different compositions. In particular, the outer membrane is relatively permeable to small molecules, while the inner membrane is much less permeable and contains integral proteins of the electron transfer chain. So, at the functions of the mitochondria. Now, mitochondria is known as the powerhouse of the cell. So, it can generate ATP from the breakdown of organic molecules. And these organic molecules can be proteins, carbohydrates, fats, etc. Now the ATP, it is the energy currency of the cell. So that's why mitochondria is the powerhouse of cell, because it is generating all this energy. Now this is how the breakdown happens. It is simplified example, that is the mitochondria in the presence of oxygen. So this is all aerobic obviously, and the organic molecules, they actually are broken down, or a digestion of these molecules happen in the matrix of the mitochondria and in the presence of oxygen, and as a result of which actually ATP gets released. Now the ATP, it's the full form of ATP is adenosine triphosphate. Moving on, to the next organelle we are going to discuss is chloroplast. Now chloroplast is more like the powerhouse for a plant cell. So it is only present in a plant cell. Chloroplast is not found in animal cells. Now the structure of the chloroplast is usually ovoid or lenticular, and they will be 2 to 5 micrometers across, whose two outer membranes, so they got two outer membranes and which enclose the luminal cytoplasm. So the luminal cytoplasm is actually the stroma, which you can see is labeled here, and it contains thylakoids in it. Now thylakoids are these like flattened stack of membranous sac-like structures, and as you can see, they look like stack of coins, and they are kind of integrated with each other, and they are kind of maybe separate or aggregate in several ways, as you can see in this model here. What are the functions of chloroplast? So like I said, the main function of chloroplast is production of ATP and oxygen, and this happens by the process of photosynthesis. Chloroplast can convert light energy into chemical energy. Chlorophyll is a pigment which is actually present in chloroplast that traps the solar energy from the sun and then produces NADPH and oxygen by photolysis of water. Carbon dioxide, which is absorbed from the air, is actually used to generate glucose. Now here is a simple picture, as you can see, of a chloroplast absorbing the solar energy and producing chemical energy in return. Now next we are going to discuss about endoplasmic reticulum, and the endoplasmic reticulum, it is like a net. It's like a, under electron microscope, it actually looks like a net, like a reticular structure. That's why that's where its name comes from, endoplasmic reticulum. And this network is actually connected to the nuclear membrane at one end and the cell membrane at the other. And this says something about the functions of endoplasmic reticulum, which we will be discussing later. Now the membrane of the endoplasmic reticulum is 50 to 60 angstroms thick, and the endoplasmic reticulum can be of two types. So there can be a rough endoplasmic reticulum or a smooth endoplasmic reticulum. Now the rough endoplasmic reticulum has ribosomes attached to the walls of it, whereas smooth endoplasmic reticulum does not have ribosomes attached to their walls. Now endoplasmic reticulum shapes can be lamellar, vesicular, or tubular. Discuss about the functions of endoplasmic reticulum. So like I said before, that why is it connected to the nuclear membrane at one end and the cell membrane at the other end? It is because one of the functions of endoplasmic reticulum is transportation throughout the cell. So it's like a like the circulatory system of the cell, kind of transporting materials throughout the cell, and that's why it is connected to the core and the outermost part of the cell. Now the exchange of molecules across the membranes of the endoplasmic reticulum, they occur by three main processes: osmosis, diffusion, and active transport. Now the next function is that smooth endoplasmic reticulum can actually synthesize phospholipids, cholesterol, and steroids by catabolism and anabolism. Now this is all lipid breakdown process. And then the next one we have is lipid metabolism. Now the next one we have is glycogenesis, and glycogenesis is the process of breakdown of glycogen, which is also another function of endoplasmic reticulum. The next function is drug detoxification. And then we have the endoplasmic reticulum. It provides an ultrastructural skeletal framework to the cell. So it provides mechanical support to the cell as well. Now the next organelle that we are going to study is Golgi apparatus or Golgi apparatus. Now the Golgi apparatus or Golgi body is actually, as you can see here, it forms from plasma membrane, nuclear envelope and endoplasmic reticulum. Now the Golgi body is composed of cisternae, vesicles and large vacuoles. Now the cisternae, they are parallelly arranged sac-like structure which are covered by smooth surface and smooth surface membrane, single membrane. And they are piled to form stacks, and these stacks can be 3 to 20 in number. Small vesicles are droplet-like sacs. They originate from cisternae by pinching off process. Large vacuoles are clear and large sac-like structures, which you can see here. All of these are marked. So there's the newly forming vesicle, which you can see in the bottom of the picture, and the vesicle is actually pinching off from the cisternae. So the cisternae, as you can see, is this kind of folded structure, like a nice kind of, if you fold like a piece of cloth and keep it folding in stacks, then this is how it's gonna look like, and that's the cisternae. And the vesicle here, as you can see, it is being formed, it's kind of pinching off from the cisternae, and that is how vesicles are formed. And then we have got some large vacuoles as well, and these vacuoles would be clear and obviously they would be sac-like. Is the function of Golgi apparatus or body. Now, it plays an important role to form the cell wall at the end of mitosis cell division. So this is a role that a Golgi apparatus plays, is the formation of the cell wall at the end of the mitosis cell division. Now, like I said, cell wall is present only in plant cells, not in animal cells. And then the next function is they help to form a new plasma membrane. So this is the function in animal cell, that they form the new plasma membrane in the animal cell, whereas in the plant cell they form the cell wall. So next they form primary lysosomes, and they take part in protein secretion. So the protein secretion is a function of Golgi body. It is responsible for neurosecretion, and they play an important role in the transport of lipids throughout the cell. So again, a transportation process which they, or a transportation function which they perform, but it is only for lipids throughout the cell. So it also releases protein molecules out of the cell, so they can secrete and also release them out of the cell. We have got the last organelle that we are going to discuss about, ribosome. Now we have already kind of mentioned it. I've already mentioned it before in the endoplasmic reticulum section, that ribosomes are found attached to the walls of the endoplasmic reticulum, and hence why there is the rough endoplasmic reticulum present. Now, ribosomes can also be found in the cytosol, that is another location of the ribosomes. Now, ribosome is a complex of RNA and protein, and hence they are known as the ribonucleoprotein. Now, ribosomes are found, like I said, in cell, either in cytosol or attached to the rough endoplasmic reticulum. The ribosome is made up of two subunits. The smaller ribosome subunit is where the mRNA binds, whereas the larger subunit is where the amino acids get added. So as you can see, the smaller subunit is the bright green smaller structure present at the lower side of the diagram here. So there's the ribosome's small subunit, it is labeled, and that is where you can see the mRNA is attached. So the mRNA is attached right to that, and then you can see the amino acid on the bigger dark green larger part of the ribosome, where the amino acid is actually getting added, and as you can see, it's a growing polypeptide chain right there. So that is the structure and the functionality of ribosome. That would be it for this study session. There will be more videos covering the differences between plant cell and animal cell in more detail, and also videos on prokaryotic cell, because we have only covered eukaryotic cell in this video. So let's do a quiz on what we have learned about animal cell and a little bit about plant cell as well. Identify the cell organelle, and we've got 10 seconds. The correct answer would be mitochondria. And here, the Golgi body or apparatus. If you liked the video, if you learned something from it, please give it a like. And if you want to stay updated for new tutoring videos on my channel study session, please click the bell icon to be notified. Thank you.